Okay, that then. Nope, that one. Okay. Okay, so like I said, the first thing we're going to talk about is the shade avoidance stem response. So thinking about in a plant, the stem cells, how they respond to daylight versus shade. And then thinking about the trade-off between elongation and defense. So you should kind of know the two flow diagrams for daylight versus shade. So that's what we can go through right now. So in daylight, what you should be thinking is there is more red light than far red light. And in shade, there's more far red light than there is red light. So as a result, in daylight, we get no stem elongation. And in shade, we get more stem elongation, which should make sense, right? If the plant is trying to reach sunlight, it's going to grow more. Okay, so what is the flow diagram for each of those? So the receptor that detects red versus far red light is called phytochrome. And phytochrome can be in two different forms, either PR or PFR. So let's look at on the left here. Can you all see my mouse, by the way? If I move my mouse? Or no?
Yeah.
Yeah? Okay, cool. Okay, so looking over here. In response to daylight, phytochrome is going to absorb red light, not far red light, and that's going to convert it to the PFR form. So the phytochrome autophosphorylates and converts to PFR. That's going to trigger a signal cascade that moves pin proteins in those stem cells to the bottom. And those pin proteins are the ones that efflux auxin. So if all the pin proteins are on the bottom, auxin is going to move out of the stem cells and you'll get no stem elongation. Instead, like we learned last unit, the auxin will actually build up in the roots and the roots will elongate. Okay, so then what about when we have the case of shade? So in shade again, we're going to get more far red light, and that's going to lead to stem elongation. So in that case, the phytochrome absorbs far red light.
Privilege of living in the US, but then shits on like our military and everything that has ever gone her like any time.
That's true.
Make sure you're muted. Okay. All right, in the shade, phytochrome absorbs far red light, and that converts it to the PR form via dephosphorylation. Then again, signal cascade happens. And now instead of pins moving to the bottom, the pins move to the sides of the stem cells. So because of that, when auxin comes down from the top of the plant, it's going to move out and kind of spread within the stem. So auxin is going to build up in the stem. Then auxin in those stem cells is going to enter the nucleus and bind to its receptor. And then the auxin plus receptor is going to bind to the Sauer gene and actually kick off its repressor. So it's going to disinhibit the Sauer gene. And then the Sauer protein inhibits phosphatase. So phosphatase is that protein that removes a phosphate group from the H pump. So if you inhibit the phosphatase, then you basically allow more H pump activity. So the H pump activity increases, ends up increasing elongation through the acid hypothesis. So by pumping out hydrogen ions, you're going to hyperpolarize the cell. When you hyperpolarize the cell, then you can activate hyperpolarization gated potassium channels. And you can also use the proton ECG to activate chloride hydrogen symporters that bring chloride into the cell. So by bringing potassium and chloride into the cell, you increase solute concentration, decreasing the solute potential of the cells, decreasing the water potential. That's going to cause water to come inside. At the same time this is happening, when you pump out hydrogen ions into the apoplast, that's going to activate proteins that will break down the cell wall and allow it to expand. So those expansin proteins will kind of break up the cell wall. So you need both water fluxing in and a breaking up of the cell wall to get elongation. But for this test, we're not so much focused on you telling us the details of what happens downstream of the hydrogen pump activity, because we kind of already tested you on elongation. What we do want you to know is kind of these flow diagrams that I've written out here. Yes, question, Prisha.
Just to confirm, when you say like for the daylight one, the auxin efflux out of the stem cells, do we need to know like where it goes after that, or?
You don't. For this test, that's not going to be relevant. But if you want to know, it goes straight down to the root cells.
Okay, got it. Thank you.
And last test, y'all got tested on the elongation in those root cells. That's not so relevant in this course. I mean, this test. Okay. So those are the two flow diagrams to know. And then you also need to know how there is a trade-off between elongating the shoot and triggering a defense response in the shade. So when it's really shady and the plant needs to elongate, but it's also facing maybe an attack from a caterpillar, it has to decide, do I want to send energy into making defense genes, or do I want to concentrate my energy on elongating and trying to reach more sun? And what we see happen is the plant most of the time decides it's more important for me to elongate than it is for me to defend myself. So how does that work? So over here in the shade, again, that's going to activate far red light and turn the phytochrome to the PR form. And somewhere in the defense pathway, this PR form of phytochrome is going to inhibit the defense pathway. So we're not quite sure. We haven't figured out where in the pathway that inhibition is happening, but we do know it's downstream of JA. So just to remind yourself about the defense pathway, because you will have to know it for this test, is in response to herbivory, maybe from some caterpillar eating, the plant is going to increase in concentration of JA. JA is going to act kind of similar to auxin, where it enters the nucleus, finds its receptor, and then JA plus its receptor is going to bind and kick off JAS. And JAS is a repressor of defense genes. So it removes the repression of defense genes, and you get more defense gene transcription happening. So what we see is in the shade, the plant can still make JA, so those concentrations can still increase, but then something downstream gets inhibited, and in the shade, we're not going to make defense gene transcription go up. So that's the trade-off you should know about. Okay, any questions about the shade avoidance response. That's pretty much it for it.
I have a question.
Yes.
For the left column, can you explain? Cause I thought auxin follows pin, but you says auxin effluxes out of the stem cell. How does that work?
Yeah, so if you imagine a stem and all the stem cells, the pin proteins are going to move to the bottom of the stem cells. So then when auxin comes in from the top, it moves through the pin proteins, which are on the bottom, and auxin just keeps moving through down, 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 down. So auxin just kind of goes straight through the shoots. In the case of the shade, the pin proteins, instead of being on the bottom, they move to the sides. So when auxin comes in, it doesn't go down through the shoot because the pin proteins aren't there anymore. It actually goes and then moves out to the side. And what that does is it keeps auxin in the shoot area. Does that make sense? Again, easier.
Yeah.
Okay.
Thank you.
You're welcome. Yeah. Truong?
Oh, I'm just wondering if we're supposed to know how the JA are synthesized and also how the PR interferes with that. Like, do we supposed to know the exact mechanism of it?
No. Nope, you should just know, like, in response to herbivory, JA gets made, and what you do need to know is what JA normally does, which it kicks off Jazz. So you need to know what Jazz is. JA, Jazz, and its relationship to defense. Those words are going to come up on the test, so you should know what they
Okay,
are,
okay.
but not the exact mechanism. You should just say, well, if it's in the PR form, then the defense genes are not going to get transcribed. Either it's going to block the JA receptor, or it's going to prevent the JA receptor from binding to Jazz, or something down there is getting interfered with. We don't know exactly what it is. Prisha?
Could you just reiterate, I guess, the difference between PR and PFR, and then like when it, like I know that it the plants, like, they absorb the red light, but then could you just explain like what the significance of converting it to PFR is?
Yeah, so phytochrome is like a receptor that detects light, and it can switch between those two different forms. And when it's in those two forms, it does a different signaling pathway. So if it converts to the PFR form, it's going to do a signal cascade that moves pins to the bottom. If it converts to the PR form, it's going to do a signal cascade that moves pins to the side. And that's why those two different forms are significant, because they tell the cell to do two different things. All right, Maya?
Yeah. So for this exam, do we have to remember like the exact mechanism by which like auxin enters the cell in the first place? Or no?
No. Remember, it's that oxalax protein, but
Yeah.
yeah, you don't need to know.
Okay, thank you.
You're welcome. Ben?
Can you hear me?
Yeah.
Okay, I had a question about the signaling cascade of PFR or PR. Does that go through the nucleus, or is that like purely in the cytoplasm?
I think technically, you don't have to know this, but technically the PFR does go to the nucleus and the PR does not.
Okay, cool. Thank you.
You're welcome. Are there other questions?
I had another question, sorry.
Yeah.
So like why can you explain why PFR autophosphorylates and PR dephosphorylates?
Why? So adding and subtracting a phosphate group is kind of like an on-off switch for proteins. Sometimes removing the phosphate turns it on, sometimes adding the phosphate turns it on. In this case, what scientists say is adding a phosphate via autophosphorylation turns it into the PFR form, which activates it. Dephosphorylating it, taking off the phosphate, turns it into the PR form, which is the inactive form. So I think what's technically correct is when you turn it into the PFR form, you activate the protein and it actively moves the PIN proteins. Whereas the PR form, you're kind of turning it off and causing it to not move the PIN proteins. So they're kind of already on the side and they get moved to the bottom if it's PFR. But if it's PR, it's off and they just kind of stay on the side. So that's the real technical.
Okay, that makes sense. Thank you.
You're welcome. Okay. So that is shade avoidance. Yeah. Cool. We can move on to water movement. Did I just stop? Yeah, stop my screen. Let's see. Nope, that's not what I wanted. Go to my next picture. Oh, that was what I wanted. Okay. So the next topic, the next topic that we have is water movement through plants. And I kind of made bullet points of the main things that you should know. So the first thing you should understand is what are the factors that influence water movement from the roots all the way up to the atmosphere. And the big picture theme of that is water moves from high water potential to low water potential. So in order to understand how water moves through, you have to understand what are the factors that influence water potential at each of these different levels that I listed. So what's the factors of soil water potential, roots, xylem, leaves, leaf air space to the atmosphere. So we'll talk about that. And then how does heat or temperature, more or less rainfall, and drought conditions change water movement by changing water potentials? So you're going to get questions on the test where I say there was hot temperature, or lots of rainfall, or little rainfall, or there was a drought. And then you're going to have to tell me how will water movement change? And your explanation is going to have to talk about what part of the water potential in each of these parts got changed. So we'll talk about how you would answer that. Then how do plant cells open and close their stomates in response to day versus night? And then also in response to drought. And then how will this affect water movement? And then also, what are the 3 hypotheses for why plants die to drought conditions? Okay, so we're going to start by what are the factors that influence water potential at each of those levels. And what I don't know why this is like moving. I'm not a computer person. Oh my gosh, what is wrong? Okay, we're going to do this. Okay. Did y'all see that? It was like moving. Oh, I'm struggling today. All right. So what influences water potential throughout the plant? And my little picture over here on the side is what you should be imagining in your head is like there's this chain of little water molecules and they're all attached to each other. And so you should imagine kind of as water's evaporating from the top, from the leaves, it's kind of pulling on a string of water throughout the plant. And kind of having that visual in your head, like the more evaporation, the more pull on that string, that's going to help you understand how water movement works. Okay, so we'll start from the top and go to the bottom. So the atmosphere or air water potential is influenced by relative humidity. That's the only part of the equation relevant. And the idea is as relative humidity increases, the relative humidity potential increases, and that causes the water potential to increase. So what does that mean for water movement? Well, if the water potential is high in the air, then water doesn't want to move in that direction because it wants to move from high to low. So increase in relative humidity means less driving force for water to move into the atmosphere, less of a gradient from the leaf air space to the air, and less transpiration. Okay, then if you go down one level, the next place we would look at is the leaf air space. So the leaf air space water potential is also just relative humidity potential. So what you should think about leaf air space water potential is that if you were to look at kind of the leaf cell apoplast, all of the apoplast has this kind of film of water along it. And that film of water is the water that actually evaporates and turns into water vapor. And because there's always that water film there, the relative humidity in the leaf airspace is pretty high, which is good because if we want it to move from the leaf airspace to the air, it needs to be high in the leaf airspace, lower in the air, and we will get that movement. And that evaporation from the leaf airspace is going to be the pull that pulls all of the water molecules in that line upward. Okay, so the next place is the leaf apoplast, which is like the leaf cell wall. And the only factor that influences it is negative pressure. Is someone unmuted? Make sure you're muted or else it distracts me. Okay, so in the leaf apoplast is just negative pressure. So remember, negative pressure is just tension on water. It's just that pull. So positive pressure we talked about inside of cells. When you push water into a cell, you increase positive pressure, you get turgor pressure. But the pressure happening outside of cells, like in the cell wall, is just due to tension, that those cohesion and adhesion forces. As you increase the tension, you increase negative pressure. So the more transpiration that's happening, the more evaporation, the more you get negative pressure. The more negative pressure, the more tension, the lower the water potential. Because as this value becomes more negative, that's going to make the water potential more negative. Okay, next we have the xylem, and it's important to realize that the xylem is continuous with the leaf apoplast. It's also continuous with the root cell apoplast. So all of these are influenced by that negative pressure. And when you get lots of evaporation, you should imagine pull to all of these areas. You're pulling it all the way up from the roots. So xylem is mostly determined by negative pressure and also gravity, but you can basically forget about gravity. It's irrelevant unless you're dealing with extremely tall plants, which we're just not on this exam. So it doesn't really matter. It's mostly just the negative pressure. So again, the more tension, the more transpiration that's happening, the lower the xylem water potential, the more pull. Okay, then we go to the roots. And again, you can think about the apoplast plus the live root cells. Water is going to travel through both of those. The apoplast is the one that's continuous with the xylem and the leaf apoplast. So as you pull on this chain, you're also going to be pulling on the root apoplast, and that's going to make more tension, more negative pressure, lower water potential. The live root cells, their water potential is just determined by what we learned last unit, the two things that influence any live plant cells: positive pressure, the turgor pressure, and the solute potential. Note that I didn't really write anything here for like how things might change, because we didn't really go over it. We didn't go over a situation where you like change the solute potential or the turgor pressure of the root cells and it changes water flow. So I didn't really write anything here, partially because you don't really have to worry about it. Okay, and then soil water potential is determined by solute potential and then negative pressure. So negative pressure here is actually not connected to the big chain. It's not due to evaporation from the leaf airspace. The negative pressure here actually has to do with how much water is in the soil. So if you increase the amount of water in the soil, you actually lower the amount of tension. So the pressure potential becomes less negative. So like we talked about in class, that has to do with the little radius of menisci. As you increase the water, the radius is going to become really big. That's low tension, less negative. So what does that mean for water movement? If there's a lot of water in the soil, then we have less negative pressure. So the water potential of the soil is pretty high, which is good if we're trying to get water to move out of the soil. If the soil water potential is high, it's going to want to move into the root cells. So if we're under drought conditions and there's not much water, then we're going to lower the amount of water in the soil. The radius of menisci is going to get smaller. That's going to create more tension. More tension equals more negative pressure, which equals lower water potential. And that's going to mean water doesn't want to leave that area as much. So there's going to be less movement of water from the soil to the root cells. Okay, any questions on the kind of the basic overview of how water moves through those cells and what parts of the equation are relevant for each section? Yes. Prisha, not with a T.
Can you explain a little bit about the difference between negative and positive pressure potential?
Yeah. Positive pressure is turgor pressure. So within live cells, as water goes in, that increases positive pressure. What does that do to water potential? It increases it. It makes it less likely water is going to move in that direction. It's positive pressure is not so relevant when you're thinking about water movement. It becomes relevant when we think about guard cells, because guard cells, when they increase their turgor pressure, increase their positive pressure, that's what causes them to kind of open the stomate. Prisha.
So,
oh, yes.
So roots are the only things that have positive pressure potential.
Roots are the only things in this diagram that has positive pressure, but really any live cells in the plant have positive pressure.
Okay, thank you.
You're welcome. Other Prisha.
Sorry, I had a quick question. Could you explain more about like what tension really means? Like, what does it mean to have tension in cells?
Yeah. So if you look at my little picture here, again, when water is evaporating, that movement of the water into water vapor is basically pulling on this chain of water molecules. And the more pull here, the more tension. And what tension literally is it's referring to like pulling on water molecules. And that happens because the water molecules have cohesion. So they hydrogen bond to each other. So these links in between them are like the water molecules holding on to each other. And as they hold on, as you get that pull, that's going to cause them to move because they're linked together. So you should
Okay.
have tension as the pull on the chain of water.
Okay, so then when you say like more negative pressure equals lower water potential, so when it says more negative pressure, that means the cells have, or like the cells are, or how does negative pressure equal more tension, I guess?
That's just what negative pressure is measuring. Negative pressure is measuring the tension. So as you get more pull, the way scientists describe that is as negative pressure.
So it's just kind of a defined thing, I guess. So if you're pulling more, does that mean like there's less pressure, which means more negative? Or would it be the opposite?
It would be as you increase tension, that's more negative pressure. And so that's more of a negative sign. So less on the pressure potential.
Okay. I guess I'll just understand like that's what they're connected to, I guess.
Yeah, it is confusing to say like more negative pressure when it is making that pressure potential value get smaller when I say more negative pressure.
Okay. Yeah, that's kind of confusing, but
Yeah.
it's fine.
Another question?
Hey, can you hear me?
I can.
Okay, so my question was about relative humidity. So, how is that, related to temperature in the air, like air temperature, and how is it like related to humidity and stuff? And how does it increase or decrease in general?
Yeah. So yeah, I would say the two things you should know about the two factors that affect relative humidity that you should know for the test is number one, temperature. As temperature increases, relative humidity decreases. So just know that.
Okay.
And then you should also know downstream how that can affect water movement. So if temperature increases and relative humidity decreases, then the water potential of the air decreases. So that's going to cause more movement in that.
So water movement towards the air from the plant,
Yeah.
basically.
Yeah.
Okay. So
And
we just need to know about temperature and nothing else
well,
for relative
temperature, and then
humidity.
also like rain and drought conditions. So if there's high
Okay.
rainfall, that's going to increase relative humidity. Lots of rain, lots of water, increased relative
Okay.
humidity, increased water
Okay.
potential. That makes it less likely that this water is going to want to leave to the atmosphere.
Okay. Okay. So you spoke about questions that we will see, like, so if there's, high temperatures or high rainfall, so this is how we approach it specific to relative humidity, right?
Yes. Yes.
And what about other things like, negative potential, negative pressure or positive pressure?
Yes. So if you see temperature or increase or decrease rainfall or drought, you're going to first start by talking about what's going on with relative
Humidity.
And how that's going to
Okay.
increase or decrease transpiration. From there, now if you increase or decrease transpiration, you're going to change the pull, right? So if you increase transpiration, you should say that's going to increase tension, which increases negative pressure potential. So that's going to cause the water potential of all of this area to decrease, and it should cause more movement through the whole plant.
Okay. Okay, yeah, okay. So,
And the opposite as well.
okay, that makes sense. Okay, thank you.
You're welcome. Other questions?
Yeah, I have a question.
Okay.
On some of the slides, I remember in lecture, that I can't find the slide, but like there was something about like the radius of the meniscus. Is that like relevant information that we need to know or not?
I think it's helpful to understand it. So yes, and just yeah, I think it would be worth knowing. So the radius of meniscus comes into play at the two ends. So at the top, the leaf apoplast, there's a bunch of like little cross links in the cell wall. And at those cross links, there's little menisci that form. And what happens is as you get increased transpiration, that's going to cause the radius of menisci to actually get smaller. And when it gets smaller, that increases tension. So if you increase tension, you increase negative pressure all the way down. So that's
Okay. So do we need to know how like cohesive and adhesive forces affect that, or just that like as it gets smaller, the tension increases?
Just as it gets smaller, the tension increases.
Okay.
Yeah,
Thank you.
I think it would be too much to ask you
Okay.
about the details past that. Yeah, and then so on the other end, the other radius of menisci was the one I was talking about in the soil. So if you imagine like two soil particles in between them, there's a radius of menisci. If there's a lot of water, it's big, low tension. That makes the soil water potential high, which is good. And then as water decreases, that makes the meniscus small, and that increases tension and makes water potential low in the soil, which is actually bad for moving water out of it. Okay, next question. Maya?
Oh, sorry. Okay. I wanted to double check drought's influence on relative humidity. So if there's more drought, that decreases relative humidity and then it increases transpiration.
Yes.
Okay. So like, but why?
Because flux reasoning, right? As you make the relative humidity really low, what do you do to the water potential there? You make it really low. If you have very low water potential out in the atmosphere, you're increasing the gradient between the leaf airspace and the atmosphere. So that's going to increase flux, make lots of transpiration.
Okay. It's just kind of confusing because like, why would the plant get rid of all of its water when it's in a drought?
Yeah. Well, that's the thing. That's why plants dry up and die to drought. That's one of the reasons. But they have realized, oh, this is a bad system we have going here. And so they've come up with ways to try to fight against that, like ABA.
Oh,
ABA
right.
comes in and says, oh, let's stop doing that. Let's close our stomates and stop that from happening.
Okay, thank you.
You're welcome. Annabelle?
Going off of that, so there's more transpiration, but there's also less water being pulled into the plant because of the smaller radius of menisci. So there's more tension and everything. So there's more going out and less coming into the plant.
And that's the cavitation hypothesis.
Okay, thank you.
Yeah. Okay. Yeah. So
I'm sorry, I just had one more question.
Yeah.
So what does ABA essentially do? Does it like stop the water loss by, allowing ions to leave. But isn't it like, deactivating the proton pump?
Yeah. Everything you said, yes.
Okay, so how does that help in like stomates closing?
So as ions
confused.
leave, what does that do to
Okay.
potential?
I
Decrease.
just remember basically saying ions flux out and some of the ions flux in.
So yeah, ions
So there's a bigger flux out.
of water. Because
Okay.
you decreased the solutes in there. Decreased solute concentration actually
Mhm.
increases solute potential, increasing
Okay.
water potential.
Water
where
potential.
Water likes to move high to low.
Therefore the gradient would increase, right?
Yes,
Okay. That makes sense. Thank you.
you're welcome. Cool. Any other questions? I'm just gonna go to the next part. I have to do this weird thing. I turn that off and then share this picture. I'm so glad I at least wrote this stuff down. Otherwise, this would have been so bad. Okay. Okay, yes. So just going over the different ways we could change water movement that will come up on the test. Temperature. If you increase temperature, you decrease relative humidity, which decreases relative humidity of the atmosphere, which decreases the water potential of the atmosphere. So then you get a higher gradient between the atmosphere and the leaf airspace, more transpiration. And then from there, you cause more tension and more negative pressure all throughout the plant. You get more water movement, which makes sense, right? If it's really hot outside, there's going to be more evaporation and the plants dry out. Okay, and then rain or wet weather. That's another one you're going to see. Again, start with the relative humidity. Relative humidity will increase with wet weather, increase relative humidity potential, increase water potential of the atmosphere, and that's going to cause less transpiration because you've decreased the gradient. So if there's less transpiration, less pull, less tension, less negative pressure, so less water movement up. Another effect of rain and wet weather is you get increased water in the soil. So as you increase the water in the soil, what do you do to the little radius of menisci of the soil particles? You make that radius larger as it fills up with water. It's just less of a circle. So that's less tension in the water. Less tension is actually less negative pressure. Less negative pressure is a higher water potential. So if you have a high water potential in the soil, that's actually a larger gradient from the soil to the root. So it makes it easier for water to go into the root. So when there's a lot of rain, you actually get less transpiration, but you do increase, at least initially, water uptake from the soil. But then there's a thing that happens where there's interaction here. So maybe you at first get the roots taking up a bunch of water from the soil because of this gradient. But eventually, as you put water into the root cells, they're not moving that water up because we don't have that pull from above. So if you don't have that pull, you don't have that negative pressure, and eventually the root water potential is actually not going to be very high. And so that movement's going to stop. Okay. And then the drought effect. So initially, a drought means low rainfall and high temperatures. So you should think low rainfall and high temps, that's going to make the relative humidity decrease. And initially, that's going to increase transpiration, and it's going to exceed root uptake, because if there's no rainfall, there's not very much water in the soil, and for all the reasons we just said, then we're not going to get water into the roots. I guess I listed that out here. So dry soil, increased negative pressure in the soil, which is going to cause a lower gradient from the soil to the roots. So initially you get high transpiration and high plant water loss when you have a drought. And then as you increase the transpiration rate and you decrease the root water uptake, what you're doing is having a kind of a hard pull from the top and not enough coming in from the bottom. And what that does is cause cavitation. And cavitation is when that chain of little water molecules breaks. So somewhere in the xylem, instead of having a continuous chain of water, somewhere along there, the water molecules broke apart. So we broke those cohesive hydrogen bonds. That's going to cause an air bubble in the xylem. And that's going to be a problem because you need this continuous chain to get movement all the way from the roots all the way up. If you break that chain, there's no way to pull all of that water up together. So that's cavitation. And it's really bad because it stops water movement through the plant. So that's one reason that plants may die from droughts. After this happens, another thing that can happen, or maybe simultaneously, is the plant will initiate a drought defense response, which is that ABA. So ABA is a hormone that gets synthesized in response to drought, and it closes the stomates. So that's going to, even though we had initially high transpiration, this will come back and say, okay, let's stop that decreased transpiration by closing the stomates, increasing resistance. And that's going to decrease the tension, the negative pressure, because we're not having water evaporate, and we're going to have less water movement through the plant. As we close those stomates by ABA, we get this other effect that may happen, which is the plants may become carbon starved. Because plants take in carbon from their stomates, from carbon dioxide. If you close them, they might not get enough carbon in, and they become carbon starved. If you don't get enough carbon in, you're decreasing photosynthesis. Photosynthesis is needed to make sugars that are then used for cellular respiration and ATP. So if you're not getting enough ATP, maybe you don't have enough energy to make, to synthesize defense responses. Maybe you don't have enough energy to activate the defense genes. So then another effect of drought is decreased defense against herbivory. So those are the three hypotheses: the cavitation effect, the carbon starvation effect, and increased insect mortality. Okay, any questions on this stuff? Feel good about water movement so far? Yes.
Do we just have to know that ABA closes the stomates, or do we need to know like what it binds to and then all of the like ions that go in and out? Like what, in how much detail do we need to know like how ABA closes the stomates?
You should know the full flow diagram for ABA, which we'll talk about. Okay. Next thing I had. Oh, I think that's next, actually. Okay. Yeah, so the next topic is how and why do plants close their stomates. And the three things you need to know is how they open and close them to daylight versus night, to drought stress, and then high CO2 environments. Okay, so let's see how they open and close it to daylight. So for daylight, they're going to do it by sensing the presence and absence of blue light. So blue light during the day opens stomates. So how does that work? We're back to phototropin. And if you're like, phototropin, what? That sounds familiar. Yeah, that was the receptor in the stem that triggered the signaling pathway to cause elongation on the opposite side of the light. But that's not what we're talking about here. Now we're talking about using phototropin to open stomates. So in response to blue light, it's going to trigger a signal cascade that activates a kinase. Remember, kinases are proteins that phosphorylate things. So the kinase will phosphorylate the hydrogen pump and increase the hydrogen pump activity. And so as we pump ions, hydrogen ions out to the apoplast, that's going to hyperpolarize the membrane and activate hyperpolarized activated potassium channels. It's like the most annoying channel to write out fully. So I just put hype activated. But that's going to move potassium ions in. At the same time, as we increase this hydrogen pump activity, more hydrogen ions out increases the gradient, and that's going to allow the hydrogen chloride symporter to move chloride ions in. So very similar to what we've seen, but now instead of sour and phosphatase, we have kinase increasing the hydrogen pump activity and then doing the same thing downstream. Okay, and then how do we open and close them to drought stress? That's the ABA response. So this is what you have to know for what ABA does. So in drought conditions, it's sensed in the roots. Roots will synthesize ABA. ABA will travel through the xylem to the leaves, to the guard cells, and they'll bind to a receptor in the guard cell cytoplasm. Then ABA plus the receptor goes into the nucleus. Oh, sorry, this is not going into the nucleus. Sorry, my brain just like went on autopilot. It actually stays in the cytoplasm, and it directly activates chloride channels, and it directly inhibits the hydrogen pump. So those kind of have two different functions. By activating chloride channels, it's going to open the channels and chloride's going to move out. And by inhibiting the hydrogen pump, we basically inhibit everything that happens over here. So we're not going to hyperpolarize the membrane, so we won't activate these potassium channels, and we also won't create a gradient of hydrogen being high outside and low inside. So we're not going to get much symporter activity either. So turning off the H pump is going to inhibit ion influx that we just talked about over here. What it's also going to do is it's going to depolarize the membrane because we're not removing those protons. If you depolarize the membrane, that's going to activate depolarization gated potassium channels. So kind of those original voltage gated potassium channels we used to talk about. And potassium is going to move out. So ABA causes chloride and potassium to move out, whereas daylight causes both of those to move in. And one thing that's really important to know is there's nothing special about any of these channels that tells the ions whether to move in or out. It all has to do with the actual electrochemical gradient for those ions. So one thing you might see is we are going to talk about these situations, but maybe we're going to change the concentrations of chloride in and out, or change the potassium concentrations in and out. And then you would have to think about, well, which direction would you expect those solutes to move given this new ECG? So if you don't feel confident about calculating the Nernst equation and doing all the basic ECG stuff, that's going to hurt you on this exam too. So we'll go over it, but that's really important to know. Okay, yeah, question?
So are we supposed to know the stuff about like too much sodium in the soil would like ruin the plant?
Too much sodium?
Yeah.
Yes, like in the context of how that would change the concentrations and then the concentration gradient. I don't know about sodium specifically. We haven't really talked about sodium, but other ions, yes, that would be hint, hint, big, important thing, definitely. But we'll talk about it. Yes. Question?
So I know that ion flux in and out is causes water flux in and out. So when basically my question is, when the water flux is out of the guard cells, where does it go?
It goes out into the apoplast, out to the extracellular space.
Okay, is that the same? So is that the same as leaf airspace?
Yes, not the leaf airspace itself, but like the leaf cell apoplast where like that water film forms. The leaf airspace is where the water becomes water vapor.
Okay, got it.
All right. The last way we get stomates opening and closing is that we talked about is high CO2 in the atmosphere. And we didn't go over a flow diagram for that one. So you should just know as you increase the amount of CO2 in the atmosphere, it seems like the plants may be closing their stomates in response to that. Yeah. Okay. So the last thing to talk about is you should know how opening and closing stomates affects all of these things here. So how will it affect carbon fixation? You close the stomates, less CO2 in, less carbon fixation. Plant temperature. As you close the stomates, you get less transpiration, so you get less evaporative cooling. So the plant temperature would go up if you close the stomates. Guard cell turgor pressure. As you close the stomates, that's decreasing guard cell turgor. And as you open the stomate, that increases turgor pressure. And then just generally water potential. So as you close the stomates, you should know specifically that that's going to decrease tension because you're decreasing that pull from the top as there's less evaporation. So the water potential of the leaf apoplast, the xylem, the root apoplast, all of that is going to go up if there's less tension and the stomates are closed. So those relationships are going to come up on the test and you should know them. Okay, that is it for that one. Any questions? Yes.
So just to be sure, does the open and closing of the stomates affect the plant temperature, or is it the other way around?
The closing of stomates affects the plant temperature by decreasing evaporative cooling if they're closed.
Oh, okay. So, okay, okay. Understand. Thank you.
All right. Then we're gonna move to the next one. Unless still have questions. Share screen. I'll share this one. Okay, dun dun dun. Everyone's favorite graph. Okay. So you should know the water potential of xylem over time graph along with the fluxes. So if I give you how the water potential of xylem is changing over time, maybe it's through a day, maybe it's over across many days. If I give you this curve, can you tell me what the fluxes are going to look like? And so that's what I drew here. So how you should interpret this is this dark line here at the bottom, that is transpiration rate, and the little dotted line here would be root uptake. So what we're looking at is first at night here, we can see the water potential of xylem is flat. And the reason for that is what I wrote here at the bottom. If there's no daylight, there's no blue light, the stomates are closed. So there's no transpiration, which is also why this line here is flat. If there's no transpiration, there's also not much tension. So the water potential of the xylem should be near 0. So I didn't label that, but this should be 0. If there's not much tension, there's also not going to be a ton of root water uptake. Maybe initially some water gets in, but if there's not pull from the top, we're not going to pull it all the way through. So not much root uptake either. And then in the morning, oops, so around 6 a.m., we get some blue light. We're going to open up our stomates. So we get an increase in the transpiration rate. And that increase is going to cause the xylem water potential to drop because we're increasing that tension, increasing negative pressure, decreasing water potential. As a result of that pull, again, remember, imagine the little chain of water. We're also going to now get increased root water uptake. But importantly, that root water uptake increase happens after the transpiration. So you can't get this to increase until we've already increased the pull from the top. And that also makes sense because if the water potential is decreasing here, we know that the transpiration rate must be higher because if you think about like mass balance, right? If you increase the rate out and it's greater than the rate in, then that's going to net decrease the amount of water in the plant. So in class, we looked at a graph of this where the root water uptake was flat. And I talked to Dr. Rusing about that, and she said when talking about this, she would be fine with either one of these explanations that I've written down here. So over time, you could say root uptake stays the same, and it actually doesn't change over time because maybe the root cells manipulate the hydrogen pumps or manipulate channels or transporters somehow to make the total amount of water going in stay the same. Or she said you could say that it will increase due to gradient. Because we're increasing the pull and increasing the negative pressure, you should see an increase in gradient and an increase in root water uptake. In my opinion, I think this is the one that's more correct. I think it doesn't make sense for it to stay the same. But she said she'll accept either answer. Okay, and then during the day, when the water potential is the lowest, that's when we have max rate of transpiration. And if it's flat and not changing, it's not going up or down, that means that the rate of root uptake and the rate of transpiration is equal. So those two lines should come together. All right, and then from 6 p.m. to 12 a.m., we're getting less blue light. So transpiration rate is going to go down as the stomates close. And that's going to be followed by a decrease in the root uptake. Why? Because again, as you decrease the pull from the top, the downstream effect of that is also going to be decreasing the pull into the roots. But if we have an increase in our water potential, that means that the rate in is higher than the rate out. That's why the root uptake is actually higher over here. And then they come back together when the stomates are closed and the rate in and out is basically negligible. Okay, questions about this graph?
What does the bottom graph mean again? Like, what is it measuring again?
The dark line here is transpiration rate, and the dotted line is root uptake, root cell uptake of water.
Why don't they overlap exactly? Why is there a little gap between them?
Because, well, first of all, whenever you see the xylem water potential graph, if it's decreasing in your head, you should think that means the transpiration rate is winning, because that means there's so much evaporation out that's causing that tension to increase and the water potential to drop. And then as you see the water potential going up, that tells you in your head transpiration is not winning. There's actually less tension happening. And so the root cell uptake is the one that's winning. So whenever the water potential of the xylem is decreasing, that's an increase in transpiration rate. Whenever it's increasing, that's a decrease in transpiration rate. And then you can think how the root, what happens with the root uptake is kind of the same thing with a delay. So first you have to get that initial pull from the top, and then that's going to affect the root cell uptake because there's more negative pressure. So this creates the initial pull, and this kind of responds to that pull because of the negative pressure, because of the increased gradient. Over here, we're getting a drop in that pull. And as a result, in response, we get a drop in the root uptake because there's less negative pressure, less tension. Does that make sense?
Yeah.
All right, any other questions? Okay, then we'll move on to the next thing.
Rachel, I'm sorry. Would you mind opening that back up just to see the bottom half really quickly again?
Yeah, sure.
Thank you.
Oops. Nope, that's not what I want to do. Yeah, and one thing, hopefully if I get my, I hope I get my pen to work, but if you guys want to like look through kind of what I've written down here, and then on Tuesday, you can maybe have digested it better and come with questions like, I don't understand what you wrote here. Can you talk about this again? That might be helpful.
Okay, thank you.
You're welcome. Okay, next one. Okay, so that's it for water movement. And now we move to nutrient uptake of plants. So there's kind of two main ways that plants take up nutrients. One is from the roots, and we just kind of focused on taking up cations and anions, and then also from the air. And I put a little star by that because you should know that most of what makes up the plant, most of the biomass of the plant is carbon from the air. Definitely know that for the test. Okay, so the main things you need to focus on is how do plants take up cation and anion nutrients from the roots? And also, how does this change in response to low nutrient soil? And then how does high CO2 environments affect nutrient uptake? So there's kind of two hypotheses that we talked about in class. So we saw, she showed some data that showed in high CO2 environments, we get less nutrient uptake. And we were like, why is that a thing? So one hypothesis is if there's a lot of CO2 in the atmosphere, you might get the situation where there's so much carbon fixation that you really increase the total amount of biomass while keeping the amount of nutrient uptake from the roots the same. And if you increase this denominator, then the total nutrient concentration decreases. So just by making more carbon stuff relative to nutrients, you technically would decrease the nutrient concentrations. The other hypotheses we said was the plants might respond to increases in CO2 by closing their stomates. And that will have two different effects. Number one, if you close the stomates, you're going to get less sap flow. Again, close stomates, now there's no movement of water through the plant. And because nutrients, they use that sap flow through the xylem to move through the plant. If you stop that sap flow, you're going to get less nutrients moving throughout the plant. Okay, the other effect would be if you close the stomates, you're going to do less photosynthesis, make less G3P, make less ATP, and that's going to cause less hydrogen pump activity. That's bad for nutrients because the way we get basically all of our nutrients into the plant is using that hydrogen pump activity to power transporters. So if you have less ATP, you're going to do less transport of cations and anions into the roots. All right, questions on this so far? Cool. Okay. Now we're going to talk about how do plants uptake anions and cations from the soil, and also if you manipulate the amount of nutrients or the ion concentrations in the soil, how does it change uptake? So we're going to kind of go through an example with potassium. So the first way we get potassium into root cells is just the same way we've always got potassium in, that y'all have already learned. So there's hydrogen pumps in the root cells, and they move hydrogen ions out. By doing that, it's going to hyperpolarize the cell. What's that going to do? Activate hyperpolarization gated potassium channels. And that's going to move potassium in. So kind of like I was saying, remember that there's nothing special about these channels that cause potassium to move in. Potassium moves in because of its electrochemical gradient. So just as a refresher, because it's important on this test, what is the ECG and how does that influence movement? So remember the Nernst equation, it gives you the concentration gradient, or in other words, it also gives you the equilibrium potential for that ion. So for most plant cells, I actually didn't do the calculation. I'm not sure how accurate this number is, but it's somewhere around here, where if you plugged in the concentrations in and out, the equilibrium for potassium would be somewhere around -80. So if the membrane potential is greater than -80, so let's say it's -50, then that's going to cause the ECG to be out. Why? Because potassium is always trying to get the cell membrane to be at its equilibrium potential. So if we're not at -80 and we're at -30, potassium is unhappy. It wants to be at -80. So potassium's over here thinking, how can I get the membrane more negative to get it to -80? We're at -30, but I want to be at -80. How can I get that to happen? And a little potassium ion says, I can move out of the cell. If me, a positive ion, moves out of the cell, that makes the membrane more negative and gets it closer to -80. So you should be able to kind of reason through all of that. That's going to be relevant for the test. If you calculate the Nernst equation, given wherever the membrane potential is, can you tell me which direction the ion is going to move? So the opposite example would be if the membrane potential is less than -80, let's say it's -130, which is actually kind of where the cell hangs out at if the hydrogen pump is active. So if the hydrogen pump is pumping protons out, it gets the membrane potential to around -130. In that case, our potassium ion is wanting to be at -80. We're not there, and it thinks, how can I move the membrane potential to be more positive? I want it to be at -80. Well, the potassium knows if it moves into the cell, it will get it closer to -80. So the electrochemical gradient is going to be inward. So that is why when we activate hydrogen pumps, potassium moves in, not because the hyperpolarization gated channels always move potassium in, but just because we're at a membrane potential that potassium wants to move in for. Why am I making a big deal out of this? Because if you change what the equilibrium potential is, oops, I didn't do that. No. If you change the equilibrium potential of the ion, it's going to change which direction it wants to move. Why would we change, or how would we change the equilibrium potential? Well, under conditions when the soil nutrient levels are not normal. So what you should expect on the test is to be given some soil that is not normal. So we've either drastically increased the concentration of potassium in the soil, or we've drastically decreased the concentration of potassium in the soil. Then you have to figure out what is the effect of that going to be using this electrochemical gradient reasoning. So if you change the concentration in or out, up or down, you're going to plug that into the Nernst equation, and it's going to give you a new equilibrium potential. And then given what that equilibrium potential is, you have to decide how is that ion going to move, given what the membrane potential is, or given if the the hydrogen pump is working. So let's give an example of that. let's say we all of a sudden make it so that potassium concentrations in the soil are super duper low. So what we've done is we've changed this value here, the concentration out. And if you make this value lower and lower and lower, what you'll do, and you'll have to do the calculation on the test, but you'll see that this number is going to get more and more and more negative. So let's say you get a value and it's negative 250. Well, if you have the hydrogen pump activity working, it only gets it to around negative 130. It never gets it past negative 200. So what is the consequence of that? In normal conditions, if you put the cell at negative 30, potassium wants to move in. But if you've changed this equilibrium potential to be negative 250, then now at negative 130, potassium actually wants to move in the opposite direction because it's trying to get the membrane potential to negative 250. How can it make it super negative? By moving out. So when you activate these hyperpolarization gated channels under these weird conditions, you don't get potassium moving in, you get it moving out. So on the test, you're going to have something like that where we're going to change the concentrations and you're going to have to interpret how the movement is going to change. Okay. The other effect of manipulating, yes, question. Sorry, it's hard when I'm just like talking and not writing. Question.
No, it's totally okay. I was just wondering if the potassium ions are moving out, would it then be like a depolarization potassium channel, or would it still be like the hyper one?
So it would be a hyperpolarization gated channel that moves potassium in, thus depolarizing the cell.
But then if like with the example that you just gave, if the potassium starts moving out to get to that negative 250, then would it just be moving out of the hyperpolarization channel, or?
Yeah, so it would be hyperpolarization gated, open those channels, potassium moves out, makes the membrane even more hyperpolarized, which would be really bad for the cell too, because the cell, it would just mess up all the ion movements. So very bad stuff happens with that. Okay. So the other effect of changing the soil, nutrient, and ion concentrations is we see changing the amount of cation and anion hydrogen symporters. So basically the plant cell can sense whether the nutrient levels are high and low, and they will make more or less symporters in response. So for example, if it detects that nitrate levels are low, then it's going to increase transcription of nitrate hydrogen symporters. If it detects that potassium concentrations are low, then it will increase making potassium hydrogen symporters. And one thing to note is this is really good, right? Because everything I just talked about in the example of making potassium concentration super low, it was really bad for the cell. The cell had a ton of problems getting potassium in under those conditions. It was actually pumping it out. But using this response, this is a way of getting potassium in, even if those concentrations are low. Why? Because as long as we have a working hydrogen pump, we can use active transport to get it in. And so the key with that is, in order to get this to work, in order to use symporters in low nutrient environments, you have to have hydrogen pumps working because those symporters won't work if we don't have the hydrogen ECG the way we want it to be. So again, if on the test I manipulate the hydrogen pumps, if I inhibit a kinase, or I say there's phosphatase activity, or I say the hydrogen pump is inhibited, that's going to prevent the plant from having this response to help it get nutrients even in low nutrient environments. Okay. Okay, I guess I did do that example. So this is just the example I talked about. So say we decrease potassium in the soil, we're going to affect the ECG by, you calculate the NERTS, you get a new equilibrium potential. That's going to cause the plant to move potassium out at negative 130. And then the other effect is it's going to increase transcription of the potassium hydrogen transporter. So you should think about these two effects whenever you're considering changes to the nutrient concentrations of the soil. All right, questions on that? Oops. Okay. Almost done. Okay. One last example with that. I just thought it might be helpful to do one more. Okay. So here's just another example in the opposite direction so that you're prepared for either direction. Let's say we have nitrate, and I tell you that all of a sudden we've changed the concentrations where it's, I don't know the units, I'm just making up a random example, 2,000 out and 1 in. So the concentration of nitrate is super high in the soil and super low inside the cell. So the effect on the ECG on the test, you'll actually have to calculate it out. So bring your calculator. But let's just say I calculated it for you. And normally the equilibrium potential for nitrate is negative 30. But under these weird conditions, now it's positive 200. So you should be able to think about what would the effect of that be? Well, under normal conditions, let's say plant's hydrogen pump makes the membrane potential negative 130. So what is that going to do if we have this new equilibrium potential? Well, before, nitrate at negative 130, how would it want to move if its equilibrium was at negative 30 and it's a negative ion? So under those conditions, actually, if you opened up like a nitrate channel, nitrate would actually want to move out. Want to move in to go from negative 130 to negative 30. Now, under these conditions, nitrate still wants to move in, right? If we're at negative 130. Oops, sorry, no. Hold on. Okay. It's a negative ion, so if it wants to become more positive, sorry, I said that completely wrong. Hold on. Let me start over. Okay, if we're at negative 130 and we're wanting to be at negative 30 and we're a negative ion, we want to make the membrane more positive. So actually nitrate is going to want to leave the cell under regular conditions, which is actually kind of typical. And so that's why we don't have like nitrate channels. That's why we have nitrate symporters, because we have to kind of actively pump it in. Okay, but under these weird conditions, where now the equilibrium potential is positive 200, if you're a negative ion and we're here and we want to be here, how are we going to move to get it to be more positive? Again, same direction. It wants to move out to make it more positive. So actually what I wrote here is wrong. So there's just going to be a much larger driving force on nitrate to leave under these conditions. So what would the real effect be? So kind of ignore what I wrote here. The real effect would be if there's a huge driving force on, nitrate to leave, we might need more energy to actually pump nitrate in. So it would require more hydrogen pumping. All right, does someone have a question? Sorry about that.
I had a question, but then I realized that you can't draw with your but like, can you just like in this example, I'm struggling to visualize what the inside of the membrane, which one's the inside? Is it the 130 or the 200?
See if I can draw.
Oh, thank you.
Yeah. This is the equilibrium potential. Oops. So of the inside of the cell. So this is what this ion wants the membrane to be at. And this is the actual membrane potential, negative 130. So both of these are more positive than this one. So nitrate wants to be at a more positive value for both of them. In order to make it more positive, nitrate is going to want to leave the cell, go out, because it's a negative ion. And that will make the membrane potential hopefully move from negative 130 closer to where it wants to be. And that driving force to move the membrane potential to these values is much bigger in this case because we're farther from where we're starting. So this change has caused an increased driving force on nitrate to leave. Yes, Prisha.
Hi, Rachel. Okay, so in this case, would we be given the negative 30 or the positive 200? Or like, would we even need Nernst for this, or how would that work?
Yeah, so the Nernst equation will allow you to calculate what these values are.
How do we do that if we have our membrane potential be like negative 130? Wouldn't like the electrical gradient then like play a role with the negative 130 of the H plus pump?
I'm confused on what your question is.
I'm just like confused on like, so how would we like see this like negative 30 suddenly become like positive 200, something like that? Like, how would we calculate that or stuff like that?
Yeah, so negative 130 is like the membrane potential. So that would be like the EG. These are not the membrane potential. These are the CG of nitrate.
Okay, and then they're not the ECG, they're the CG, and then you combine the negative 130 and the CG to get the ECG.
Yes, yeah.
Okay,
so what
perfect.
You find
that
is,
makes more sense.
Okay, the gradient is going to be much higher in this case.
Yeah, cool, perfect. Thank you.
You're welcome. And then quickly, I just want to say for this too, I said you should think about the ECG effect, how it's going to change the gradient. and then also the transporter effect. So if we make the gradient even bigger and even less likely to get nitrate in, the cell is going to sense that and it's going to upregulate the symport. So the opposite of what I said, because when I was doing this, I got it wrong. Here. There is a huge increased gradient, but it's an efflux. And because there's a gradient of nitrate to move out, it's going to upregulate that. Hopefully that makes sense. I'm sorry, this is wrong. Yeah. Question?
Yeah, can you hear me?
Yeah.
Okay, so I was just a little bit confused on how you got two numbers from using the Ernst equation for the concentration gradient, because when I've done it before, I've only gotten one number. Are you saying that there might be like an extra step that we have to do after the Ernst equation to like get the positive 200?
No.
Or is it?
No, this is just me being bad at teaching. So what I'm trying to show here is, I'll say like under normal conditions, the concentration of nitrate in and out is this. So using the in and out, you would put into the Nernst and get a value. And so under normal conditions, I'm saying the value you would have gotten is 30, negative 30. So that is the value of the Nernst equation, the concentration gradient under normal conditions. Then my next sentence would be, but oh no, we're in some weird soil where now the concentration of nitrate in the soil is super big. It's like 2000 and it's 1 inside the cell. And then you plug those two values in to your Nernst equation. Oops. And I'm saying you got that.
Oh, okay, that makes more sense. Thank you.
You're welcome. Other questions? I feel like maybe in this section I might have confused y'all more.
So that value helps us come to the explanation we just spoke about, right?
Yeah, so basically what you're gonna do, if I could give you like a step-by-step, you're gonna calculate the ECG under normal conditions. Then you're gonna calculate it under the weird conditions, either much lower concentration in the soil or much higher.
Okay.
Okay. And then you're going to think about how the ion movement is going to change under normal conditions versus these new weird conditions.
All right.
So this is all stuff we did in like the neuro section, kind of.
Yeah, okay.
So you'll talk about how it's going to change the movement, change the gradients. And then on the other side, you will have to think about how it'll affect the transporters. And
okay.
If it's harder to get the ion in, then we're going to upregulate the transport. If it's easier to get the ion in, then we'll downregulate the transport because we need less.
All right, that makes sense. Thank you. Thank you so much.
You're welcome. Okay. Any other questions about this? This was confusing. I will draw it out so pretty on Tuesday. All right. Then we get to our last one. This is my last page and we're done. Okay. And hopefully I didn't write anything incorrectly on here. We'll find out. Okay. Photoperiodism and flowering. Okay, so there's basically three. Oh, question?
Oh yeah, sorry. I was just gonna ask what the plan is for Tuesday. Are we gonna be just going through some practice problems, or would it be generally like going over this?
Yeah. I don't know. You know what I'll do is I'll send out a little poll and see what kind of form y'all want or what most people want.
Okay, thank you.
You're welcome. Okay, So photoperiodism and flowering, there's three facts you need to know, and then you'll get any question correct. What are those 3 points? Number 1, the coincidence of light, specifically red light that triggers the PFR form, and a peak of CO mRNA, the coincidence of those 2 things happening, that equals stabilization of the CO protein. So you need both light and phytochrome in the PFR form, and you need a peak in the CO mRNA. And if both of those 2 things happen, you get the CO protein sticking around. So how does CO mRNA change throughout the day? It kind of looks like this graph I have at the bottom where there's like a peak in the afternoon. So you need red light here to happen in order to trigger the PFR form, and that stabilizes the CO protein. If this doesn't happen, you'll get no CO protein. Okay, then if the CO protein is stabilized and it's around, it does 2 different things depending on if it's a long day plant or a short day plant. So if it's a long day plant, the CO protein is an activator of the gene FT. And the gene FT is florigen, and it's basically genes that trigger flowering. So CO protein is going to trigger flowering in long day plants. In short day plants, there's the mistake. I think I wrote a P. Short day plants, not short P plants. Short day plants. The CO protein is a repressor. So if the CO protein is stabilized and it's around, it's going to repress the transcription of FT, which is florigen, so you get no flowering if the CO protein's around. Oh, so I guess I just combined that last point. So if it's made, the FT protein is a hormone that triggers flowering, and because it's a hormone, it travels across plant tissue. So it's actually made in the leaves, and then it travels to the meristem. But this is also a key point because if one part of the plant is making FT, it can activate flowering in a grafted part of another plant. So if you have like a long day plant that is making FT and you graft on a short day plant that's not making FT, you can cause that short day plant to still flower because the FT hormone can travel to it. Okay, so if you know those 3 things, you can answer any question about an experiment on flowering, which is what you're going to get. You're going to get some kind of experiment on flowering that looks like this. So I made 2 examples for you that are similar to what you'll have to do. Okay, so you'll be given some information about when the CO mRNA peaks. So let's just say it says in the afternoon at this time. That's when the CO mRNA peaks. And then we do these 2 experiments, A and B. In experiment A, we have light here and then dark all through this period. In experiment B, we have light here, dark, and then a flash of light right here, and then dark. And then what we're going to say is, tell me whether flowering occurs or does not occur for these 2 different types of plants.
Like which equation he is for. Yeah, so easy change it to get how they're
Mute yourself, please. Okay. Okay, so for experiment A, let's say we have a long day plant and a short day plant grafted to it. So under experiment A conditions, we have basically dark throughout the peak section. So what you should think is that means there's no red light. No red light is happening here. So there's no PFR form being triggered in phytochrome. So we're actually just staying in the PR form. And the PR form is not going to stabilize the CO protein. So what that means is in the long day plant, there's no CO protein and there's no activation of FT. Okay, what about the little short day plant we grafted on there? So in the short day plant, again, there's no red light. So there's no PFR form, and it's in the PR form. So there's no CO protein in the short day plant either. But this means something different. If there's no CO around in the short day plant, there's no repression of FT. So in that plant, FT gets made. And if FT is made, it can travel to the long day plant. And what you would expect is, under this condition, you would get flowering in both parts. Questions on A? All right. Experiment B. So now we have a short day plant with a long day plant grafted to it. And here we have a little flash of light right at the peak of the CO mRNA. So that means that there is red light at the key time. Oops, my flux is on. Let me disable that. I don't know if y'all can see it. So we have red light at the peak, which means we're going to get the PFR form, and we're going to stabilize CO in both plants. So for stabilization of CO in the short day plant, that's going to mean we activated the repression of FT. If we have a stabilized CO in the long day plant, that's going to activate a transcription factor activator of FT. So we'll transcribe FT, we'll get flowering in the long day plant, and because FT is a hormone, it can travel over to the short day plant and cause flowering there too. So both experiments would result in flowering throughout all of the plant. Yes, question?
Does it matter when the flash of light occurs, or like if it would happen in like the late afternoon when it was decreasing, would that like decrease the chance of it flowering?
Yes. So it specifically has to occur at the peak of the CO mRNA. And if it's not at the peak, it's not going to work.
Okay, thank you.
You're welcome.
Rachel, so if anytime we're grafting like opposite day plants, so like long day plant and a short day plant, like as long as they're opposites, do we always assume that flowering is going to happen for it because FT is made in at least one of the plants?
Yes. Yeah. I'm trying to think of a situation where you would get like no activation of FT in both of them at the same time. And I can't think of how that would occur. So yeah, I think that would be safe to assume.
Okay, thank you. And then also, I'm just curious, but this might be completely irrelevant, but is there like ever a thing where like it's daylight and there's like a flash of darkness, and does that change? Or I don't know, maybe that's like completely irrelevant to what we're learning though.
No, yeah, that is. So fair game for this would be, I could give you a flash of white light, and white light you should assume has more red than far red. I could do a flash of darkness, which would trigger changing back to the PR form. So no stabilization of CO. I could flash far red light, which again, PR form, no CO. I could flash darkness. Yeah, all of the flashes are possible. And you just need to know, is that going to make a PR or PFR form? And then figure out downstream what happens.
Okay, thank you so much.
You're welcome. All right, any other questions so far? I'm sorry this was not as great as I wanted it to be. Yes, question.
Hi, yes. So, to my understanding about flowering in short day plants, I was wondering how that happens when I thought your CO represses the transcription factor of FT in short day plants. So, how exactly does flowering occur in short day plants?
Yeah, so if the CO protein represses flowering, then the absence of CO protein causes flowering. So darkness doesn't activate their repressor, which is the CO protein, and then they get flowering. So they like lots of darkness, which is why they're short day plants.
Okay, yeah, that makes sense. Thank you.
You're welcome. Other questions? This is all I have, so you can ask about anything. All right, well then, Tuesday, I promise I'll have my pin working and I'll send out a poll so y'all can tell me how you want me to structure it or tell me what sucked about this one and what I could do better. And then ask on Piazza questions that you have. And yeah, I can also, I don't know, I'll post the recording and do whatever else is helpful to you guys. So that's it.
Thank you.
Thank you.
Thank
you.
Thank you very much.
Of course.
Thank you.
Oh, Rachel, I just had a quick question. Sorry. It was like mentioned really briefly at the end of class lecture, but do we need to know like how G3P, like, because we were looking at what the energy, like where the energy and matter goes when like G3P is broken down. Do we need to know the specifics of that, or like will we need to know anything about that? In terms of like how G3P energy and matter is converted into making like proteins for the plant?
No.
Okay.
Yeah. I know, I was surprised too, because I feel like we spent a long time on it on class, but no.
Okay, perfect. Thank you so much.
Yeah, another question? No. All right.